So who's heard of molecular genetics? I think everybody would have heard of molecular genetics. Two of the biggest things that have happened in my career in the 30 years that I've been working in Rust, in research, one would be email, which is probably not such a good thing. The other would be molecular genetics. These are probably the two most revolutionary things that have happened during my career. But the point I would make is they're both tools. That's all they are. Email's a tool for communication. Molecular genetics is a tool that helps us do our work, I suppose, adds a little refinement and allows us to do things a bit quicker. So it's important to remember molecular genetics, molecular biology is not the be-all and the end-all. And in fact, in the absence of biology, the basic biology we've been teaching you, the phenotyping, identifying races, without that, molecular genetics is nothing, right? It's just a tool. And I have to tell my students that. Often I get people come to do a PhD with us or postgrad, and I say, so what do you want to do? And they say, I want to do DNA work. I want to do molecular biology. You know, and I have to tell them, well, that's fine, and it's important to know that, but if you don't understand the basic Rust biology, it's not going to really help you. All right, so just in the lecture that we've got today, and really 1 hour is not enough time to cover the whole field of molecular biology. So, so this is a fairly general overview of the technology with some examples of how we can actually apply it as a tool to help us with controlling Rust. So, molecular genetics and DNA marker technologies, a little bit of history on that. Rust resistance gene mapping using markers. DNA markers in wheat, breeding for resistance, Rust resistance gene cloning. So this is the possibility that we have now of using transgenic approaches to controlling Rust. And then finally, how we can use DNA markers in Rust pathogens to help us with the surveillance work we're doing. All right, so molecular genetics, there's a definition of it there, the structure and function of genes at the molecular level. And really this goes right back to the discovery of the double helix by Watson and Crick way back in 1953. It was really that started this whole field of research off. One of the key discoveries was or developments was the development of the polymerase chain reaction. Everyone's heard of PCR? Yep. This was very, very ingenious invention by a guy called Carey Mullis, who won the Nobel Prize in Chemistry in 1993 for the work that he did in developing the polymerase chain reaction, which really was a quantum leap in all of the DNA work that we do. You go into any lab these days, you'll usually see a PCR machine sitting somewhere on a bench. What it does is it allows you to artificially make DNA molecules in the lab from very, very small amounts of template DNA. And of course, what we've used that for many, many different things, but it's allowed us to develop DNA markers linked to important traits such as resistance genes, okay? And I'll give you an example of this during the presentation. So that's one of the most important applications of this technology. So you all said you've heard of PCR. Do you know how it actually works? Right, basically you know that DNA is double-stranded, right? The first step, you have to mix it with what we call a primer that starts the building of the new piece of DNA, and then these nucleotides. And the first step is that you heat that up in the machine and the two strands separate. You then cool it down. The primer comes along and anneals to each of the strands at a specific point that it's designed to anneal to, and then DNA polymerase makes a new strand of DNA, and then you just can repeat that cycle over and over again, and you get an exponential increase in the number of fragments you're generating. So it's just a heating and a cooling process. Every time you heat, the DNA denatures into two strands. Every time you cool, the DNA polymerase enzyme makes a complementary strand, so you go from 1 to 2 to 4 to 8 to 16 to 32, and it's exponential in copies of the DNA. And I think typically in a PCR reaction you do about 40 cycles. Does that sound right, Devinder? Around about 40. So you're amplifying up the DNA, you're making new bits of DNA each time you do a cycle. But the important thing that I wanted to point out there is these primers, and I'll be talking about these in the talk. These are what actually are specific to bits of DNA, and that's what actually starts the manufacturing of the new piece of DNA each time you go through a cycle of PCR.